On this special year-end episode of Translating Proteomics, hosts Parag, Mallick and Andreas Huhmer discuss 3 of their favorite proteomics publications from 2024. They'll cover 1 paper in each of the following topic areas: proteomics in preclinical research, proteomics in basic research, and technology development in proteomics. Now here are your hosts, Parag and Andreas.
Hey everyone, welcome to a special year-end episode of Translating Proteomics. On this episode, we'll discuss 3 papers from 2024 that we believe are great examples of preclinical research, basic research, and technology development in proteomics. Of course, there are many other great examples of proteomic researchers out there, and particularly out this year. We'd love to hear about some of your favorites. Please share your favorite proteomics publications from 2024 in the comments, and we may cover them in a future episode. The first paper we'll be discussing comes from the lab of Professor Bernhard Kuster at the Technical University of Munich. Its lead authors are PhD students Stephen Eckhart and Nicole Berner, and the paper is titled Decrypting the Molecular Basis of Cellular Drug Phenotypes by Dose-Resolved Expression Proteomics. This particular paper is an amazing example of preclinical studies. In it, the researchers began by looking at one particular cell line, Jurkat cells, and studying it across literally 144 different therapeutics across a range of treatment conditions, different doses, and then examined how all of those different variations impacted the proteomes. This was a massive, massive study, looking at thousands of proteins per condition and really trying to understand which proteins were influenced by which drugs. I found this paper so exciting, in part for, one, its scale. This was a massive, massive undertaking by the authors. And these authors have in the past been critical drivers of many other large-scale studies, including an analysis of the NCI60 cell lines, as well as the first draft human proteome. Beyond that scale, though, what was really exciting for me was that they really were using dose and dose response as part of their interpretation of proteome response. This is a critical driver of understanding what proteomic changes are really indicated and driven by the therapeutics, and our dose-response relationships are unlikely to occur at random. Whereas if we look at things like simple A-B comparisons, it's possible that you might see something that is high abundance or low abundance just at random. But seeing a trend that tracks with dose, on the other hand, is incredibly rare and incredibly special. And so for me, it really was the combination of the looking at a response that was correlated with an alternate variable, as well as the scale that made this such a special paper.
Yeah, I think what I liked about the paper is that it is one of the examples of not only large scale, but also with a multiomics aspect of it. So what they also looked into is, what if we compare the protein expression levels that we measure with the mRNA levels? And it turns out, as you would probably expect by now, where many of these studies have been done, that mRNA does not necessarily correlate with proteins. And what was interesting, though, is that some effects of some drugs, actually mRNA and protein level measurements were just the same. In some cases, it was completely different, where you got a lot of mRNA responses, but that was not reflected ultimately in the proteome. And so that actually highlights how important the direct measurement of these effects are in the proteome itself. And what is also different in this paper than in many other preclinical type mechanism of action study is that they don't only look at one data point, as you say, they look at the entire dose response curve. And so they get a much better idea of what happens not only to the target, but what happens, in subsequent pathways. And what is most important, and the authors state that at the very end of the paper repeatedly, this is measuring essentially the end state of a cell. So once the cell has been exposed through a series of different drug doses, how does the cell respond and what's the end state of the cell? So that is very interesting because it goes way beyond just estimating what happens to the target. I mean, you do a typical mechanism of action study where you just study the target that you think the drug has and learn from that. But now you have visibility to what are secondary or tertiary effects of the drug treatment. So I thought it was very interesting. Another reason I think to pick the paper is that all of the data is actually available. So you can go and dive deeper into particular either drugs or particular protein groups and learn more about how they can be effectively drugged or targeted. So I thought that was very important these days that actually, the data that the lab generated is publicly available. And a comment you made, Parag, was that, it's a massive study. So they literally had the study going on for an entire month on a particular instrument. And what was interesting to see that typically LC-MS-based instrumentation doesn't, work flawlessly for a month. But in this case, they choose to essentially put in a gas phase filtering device, a FAMES device, and use the higher flow rate to really make the study work over such a period of time. And nevertheless, they got pretty good protein coverage for about 8,000 proteins. So overall, a major work, as you say, but with, incredible insights.
I think one of the other key aspects of the work was that in addition to it being a multi-omics study, they did a lot of work on the computational side, developing a tool to specifically ask which proteins have a pattern of changes in abundance that maps to the dose response. And as I mentioned earlier, they looked at 144 drugs, 16 different classes of drugs, and they treated for 18 hours with 5 different doses in steps between 1 nanomolar and 10,000 nanomolar. They also looked at a variety of different negative controlled treatments as well. And so from an experimental design standpoint, that was exciting. But because they had these 5 steps of treatment, they were then able to say, all right, well, which proteins had an arbitrary pattern, which ones had no pattern, which ones could they then deconvolve and say, all right, this one actually has a distinctive pattern that correlates very specifically with dose. And for me, one of the most interesting results of the study, perhaps not surprising in hindsight, was how few proteins changed. Now, that's something that when we think we're dumping 10,000 nanomolar of a drug onto a cell, we would expect that there would be non-specific cell death and lots of just general cell freaking out, which would lead to massive proteome rearrangement. But instead, I believe it was less than 20% of proteins were changing in many of these systems.
Correct. Yeah, almost the 80-20 rule applied here, right? I was actually surprised, as you were, that the actual proteins affected by each of those treatments was, about 20% of the proteome. I think, that tells us again that the cell has to, manage, any type of external stimuli with, with a limited budget of resources that it has, and you can't just wildly start, regulating proteins. And this is where maybe the other aspect of the paper, which is interesting, should be mentioned here. They actually do mention that PTM, it would be great to have PTMs included. They included not a full set of PTMs, but they measured some methylation to look at particular T cell activation example that they showed in the paper. But in follow-up experiments, one could imagine that you could do much more phosphorylation or acetylation or other PTM-specific signaling work with that in conjunction with the drug treatment. And that obviously would create a different dimension of insights. How do these drug binding essentially create different pathways or signaling pathways? And obviously with the more modern next generation drugs, the degraders emerging, we probably would want to couple this with a isotopic labeling study where you can actually look at not only at the the protein level, but also at the degradation of proteins. And in fact, the authors mentioned that there would be an additional improvement on what they have done.
Yeah, that's right. So the study doesn't really get into the dynamics of the proteome. So when we think about some of the potential limitations of the study, one, of course, was that they used Jurkat cells for everything. Number two was that while they did a dose response, they didn't do a time response. Number three was they didn't have an ability to look at changes in translation or degradation. They only were able to look at absolute levels, so they couldn't look to understand what the genesis of that was. I think for me, one of the most important extensions really would be to look in different model systems. We know that targeted therapies specifically only work in a small percentage of cells or people who have activating mutations, for instance. And so that sort of fine resolution detail wouldn't be able to come out from a study of Jurkat cells. And in fact, some of these drugs that are, for instance, histone-targeted drugs or general DNA damage drugs, the timescales of response may also not be captured in the 18-hour time frame that they're looking. One of the signaling kinase inhibitors, for instance, they may actually operate on the PTM level very, very, very quickly. Within minutes even, you might see changes in the phosphorylation of proteins, but the changes in protein abundance may have to propagate all the way down to changes in transcription before they then propagate up to translation and changes in abundance, unless they're specifically altering post-translational degradation cascades.
Correct. Yeah, I think it's a paper worthwhile reading, and I think we're looking forward to the next or additional work from this laboratory, utilizing the proteome wide scale technologies to really look at holistic responses on drug treatments and drug targeting.
Yeah, I think one thing for us to think about is there have been a few other studies that have looked at drug response in using proteomics. So probably one of the larger ones is the Cancer Cell Line Encyclopedia study that was published a few years ago. And in that study, they looked at a much smaller set of drugs. They looked at a larger set of cell lines, but only typically one dose. And so I'm curious, Andreas, from your perspective, how important is the dose response aspect of this study?
Yeah, I mean, they go into details, actually, how they filter out those data that they obtain by matching a sigmoidal curve on it, where which is a very well established way of looking at drug binding. But they also admit that there's many other drug mechanisms that would probably manifest itself in a different pattern, maybe in just a step function, turning off, turning on type pattern. They did not look into that, and they basically made the data set available so others could use more sophisticated data analysis approaches to look in some other signaling events that happened. And so it's a great resource, and I wonder with machine learning emerging, that if you combine the study you mentioned, maybe some of the other studies, single dose studies, but that they use, for example, a melting curve type approach, a temperature change approach to modulate the binding. Maybe there is a basic data set that can be used for machine learning and really understanding how these particular drugs act on a whole proteome.
Just following up on that, one of the things that I find really interesting about their result is to what extent does the protein that is being targeted by the drug actually change abundance? And the answer in general was not, that almost all of the changes were seen around that protein, downstream of that protein. And I wonder, as we think about developing therapeutics, as we think about toxicity, off-target effects, downstream consequences, how does this study inform our thinking when we're trying to develop that next therapeutic, when we're trying to find that next drug target?
Correct. And I think this is where, hopefully a lot of adoption of this type of approach is going to give us more insights in the future.
So do you see that this is the way that all proteomic drug screening studies are going to be done in the future at this sort of scale and with everything being done in multiple doses and times?
I mean, if you look at the ultimate cost of a study like this, I think it's not prohibitive in thinking about that. Many of the studies that are done today could be probably enhanced or replaced by a comprehensive study like that, and very quickly learn for a particular drug class, what kind of other effects should we expect in a proteome or in the cellular model. Then obviously could be followed up later on in organisms or other model systems. But clearly, it's not so cost prohibitive or time prohibitive that you would not want to include that as a basic screen for every drug target that you're developing.
One thing that they particularly called out is that relative to the chemical biology methods that are aimed at identifying specific targets of drugs, specific direct targets of drugs, they were explicitly not doing that. They were not trying to identify what proteins on target or off target were bound by these therapeutics. How do you think you might combine one of those chemical biology data sets where you actually knew the targets alongside a study like this?
Interesting. If you keep in mind that they state that basically their intention was to capture the proteome after the treatment, when it has essentially reached a different stage. In that stage, probably is a new proteome that then has a different reaction function. One could think of repeating these experiments with another set of doses or a different drug, to see how that proteome continues to respond to additional treatment. In that sense, I think because it measures more dynamics than the single dose studies, I think these studies are probably much more important to do than maybe combining the single dose study with a dynamic study like that.
One of the arguments that often is raised in studies like this is that you're looking at the cell so far after the treatment that essentially you're looking at the house after it's already burnt down and trying to figure out what started the fire, which is certainly something that can be done, but is challenging. If we really wanted to understand the cascade of events and potentially feedback loops, how might we have to extend studies like this in order to capture that fine-grained detail?
Good question, but I think there's also many other aspects of it that would have to be, considered. This is now a cell model, which we know does not necessarily replicate what's happening in real tissue. So maybe the better way to do this is to repeat this in, let's say, organoids or other, tissue resolved model systems. I think the cellular study was probably very, got good first step to sort of understand what happens if you have a very reproducible environment. But I think ultimately this would have to be reproduced in a tissue level type analysis.
So maybe just to sum up, this is a really remarkable paper setting forth basically an entirely new paradigm for large scale proteomics of investigating therapeutic response and amongst the key findings really were that the scale of proteome rearrangement was maybe smaller than we anticipated, and that there were cascades that went far beyond, much further throughout the cell than I think were anticipated. Overall, this is a foundational study and an exciting one to build on for the future.
Yeah, congratulations to the authors.
Obviously, this is a really important paper, so much to think about. We'd love to hear your thoughts on this paper, where they can take it next. Please let us know in the comments.
So next up, we have the paper entitled Natural Proteome Diversity Links Aneuploidy Tolerance to Protein Turnover from the labs of Professor Judith Berman at Tel Aviv University and Professor Markus Ralser at the Charité Universitätsmedizin Berlin. The lead authors were Julia Münzner and Pauline Trebule, both postdocs in the Ralser lab. So in this paper, the authors studied did an integrated genomic, transcriptomic, and proteomic study from about 800 natural yeast isolates, where they studied the effect of aneuploidy and euploidy from natural as well as lab lysates. And what they found in principle is that in natural yeast cell lines, the uneven chromosome number is relatively well compensated and does not have effect of the fitness function. Whereas in lab-generated aneuploidy yeast cell lines, the actual effect on the proteome was very, very different. What they did is they used protein turnover rates with pulse labeling and stable amino acid isotopes to provide a precise level of proteins for these, proteins in the for these lysates from aneuploidy chromosome lysates, and then looked at the degradation rate to find out exactly how the cell dealt with the excess protein expressed in the particular yeast. Previously, it was assumed, or it was shown, that when the cells had an uneven number of chromosomes, the additional expression of proteins was primarily controlled by, for proteins in large complexes, where the smaller protein complex members were rapidly degraded. In this particular paper, they show that this is not the case. In fact, that in the natural lysates, the control of the proteins were much wider distributed across the proteome than just for proteins in protein complexes. What else was interesting in the paper was that they actually used a fairly comprehensive multiomics approach to make sure that they actually had sufficient evidence to look at what level of protein comes from the actual synthesis, as well as what actually is the protein that was degraded.
One of the things that I love about this paper is that they're doing a really fundamental deep dive into the biology of cellular regulation. And one of the key authors on this manuscript is Matthias Selbach, who is known for his really legendary paper looking at protein translation and how it relates to RNA transcription in eukaryotic cells. And I think that foundation is clearly evident here. Really asking the question, when we have copy number changes as a result of aneuploidy, what happens? Do we just end up with a whole lot of excess of protein? Do we end up with the cells just freaking out and dying? Do we end up with compensation mechanisms because less protein gets made and there's transcriptional gates? Or, as they showed here, in some instances, in some very specific instances, you have changes in degradation. And what was really important about this is that that question was very much an open question, and the aspect of how this changes in made cells versus naturally occurring cells was perhaps the single most important finding that they had. When aneuploidy exists, it can cause strain on the cells because they have to do all this extra work in order to make extra things, and the regulation may be not as tightly controlled. On the other hand, in naturally occurring cells, there's been an evolutionary process that perhaps has culled those cells that are unable to compensate for this genetic aberration. And so the clearest finding in this paper was that they saw significant attenuation driven by post-translational degradation in naturally occurring cells, much more so than in created cells. It was a really unsubtle difference. It was a difference of almost 150%.
Yeah, and I think the authors also mentioned that in particular lab lysates or lab strains, they think maybe the actual evolution to adapt to these changes was not completed, whereas in the natural lysates, you would assume that adaptive forces had now at this point sort of traded off the extra work and the extra energy needed to degrade these higher levels of proteins. And so I think what I took away from that study was that when we look at all of these protein expression studies in cells that are grown for generations in laboratories, we should be careful of the conclusions that we have, because there may be mechanisms that in natural environment be adaptive. In a human body, the cells may have adapted after a while to a drug treatment, for example, connecting it to a previous conversation, while that might not be possible in cell lines that are essentially grown in a laboratory.
One of the things that I'm really curious about as a consequence of this paper, so this paper was done in yeast, where it's very clear you can get chromosomal duplication and those forms of genetic alteration. How do you think this applies to eukaryotic systems, human systems, where you have things like copy number polymorphisms? It's clearly a little bit different than aneuploidy, but on the other hand, you are having more copies of a gene. And we've generally seen that with more copies of a gene, you get increased in transcription, but we haven't necessarily seen that strong a correlation between copy number variation and increases in protein abundance. Do you think that the mechanism that they're showing here of the sort of, even if the rates of translation are kept the same, the amount of transcription increasing would have led to more protein abundance, except that there's a switch in the degradation machinery switching on also in human and eukaryotic cells?
It's possible. I would suggest that, the environment in a natural tissue in an organism is just much more different, because you're much more constrained in your input factors, whether that's, the oxygen in a human body, or the nutrition. I guess in these cultures, the nutrients are never really a limiting factor, and that might allow the cell to escape these type of unnatural or imbalanced conditions rather quickly, where that might be very difficult for a particular tissue in an organism. I think what would be interesting to study is if you can actually put together several cell types in, let's say, a small model organism or an organoid, and then start studying this in the context of a tissue.
That's interesting. So in terms of implications, because this was done in a purely in vitro setting, it's possible that the resource constraints that don't exist here and do exist in a human system may lead the regulation to be different. So you might, for instance, see greater regulation at the transcriptional scale than they observed here.
Yeah, I mean, the question to ask obviously is, how does the cell know, and I put the know in quoted commas, right, that with the extra production of a particular protein or many proteins, it actually has to put in work to regulate those, right? There must be some higher level control mechanism available. And if there's none, then typically it's the availability of nutrients that controls which particular process is prioritized in the cell. So I would think that if it would be as easy controlling protein expression, we would probably found some kind of switch already in let's say cancer cells, where uncontrolled cell proliferation is a big problem, and if such a high level switch would actually exist, other than for example nutrient availability, we will be able to target those. So I think what's interesting that basically there is a fairly dynamic mechanism available in the cell that immediately starts degrading the excess protein, particularly if there is energy sufficiently available to do that, and immediately come back to some kind of equilibrium. And it's interesting to sort of think about what is that driving force in the cell that actually, brings the cell back to this equilibrium of the right copy number for each of the proteins.
That's interesting. There was a study that was done many years ago by Scott Manalis's lab at MIT. They built a sensor that could literally weigh the mass of individual cells. And what they found is that the cell division was a very tightly controlled process, where as the mass of the cell expanded, it hit this almost critical juncture, and then all of a sudden the cell would initiate division. And so there clearly is a sensing mechanism inside the cell for just knowing how much total stuff it has. And so I wonder how tightly it's taking advantage of that to regulate broad processes throughout the cell versus particularly narrow axes like replication or metabolism.
Yeah, correct. So maybe as a conclusion from this, very important paper again, which is a continuation of early work that was done in this area by Matthias Silbach, as you mentioned. Another great paper on how the cells actually control various mechanisms to actually maintain some equilibrium around protein content of the cell. And so a great study that hopefully many people read, and in future, hopefully people can build on that in different cell types and maybe even tissues.
Yeah, definitely in the future. This was done in yeast. It was done in one particular strain of yeast, looking at eukaryotic systems grown in different environments, grown in environments that are nutrient restrictive or mechanically restrictive, and seeing to what extent do these findings generalize. But overall, a really detailed, remarkable study looking at hundreds of different strains of yeast with and without aneuploidy, synthetically created or naturally occurring, and really bringing all of it together to understand how there exists an extra layer of evolution and regulation that occurs in naturally, or that is found in naturally occurring systems that are not sufficiently recapitulated in artificial models. And this certainly is something that I had never contemplated, that as we're modifying some of these genetic variations, the extent to regulatory changes may be much more far-reaching than maybe we anticipated.
Yeah, so some people are often stuck with the number of proteins covered in some of these studies. Do you really think, Parag, that that the coverage of the proteome is very critical there? Do we really need every single protein in the proteome covered by these type of studies?
I was really thrilled with this study. Yes, they didn't measure every single protein in yeast, but they really focused on the biology, and they found an important biological insight. And they focused very specifically on how can we ask and answer this important biological question? How can we use proteomics and its toolbox to do that? And so I was really excited that they got beyond the numbers game, and they weren't focused on, can I measure 10 more proteins? They were instead focused on how can I drive insight?
Agreed. Yeah, this was a great discussion of a paper that dive deep into the basic biology of how yeast controls the protein levels. If you have similar papers or want us to discuss a paper of interest, please let us know in the comment section, and we'll give this paper a look.
For our last highlighted paper of 2024, we have an amazing example of technology development in the field of proteomics with a paper titled Multi-Pass Single Molecule Nanopore Reading of Long Protein Strands from Professor Jeff Navala's lab at the University of Washington. The lead authors are Keisuke Motone, now an assistant professor at Osaka University, and Daphne Kontorgiorgos Heinz, a PhD student at the University of Washington. Daphne, I apologize for butchering your name. In this work, the authors developed a new nanopore-based approach for measuring protein sequences. And this work is really notable because it extends the previous work of Case Decker and colleagues that were able to do multi-pass reading through a protein nanopore. They added a specific advantage that they used an unfoldase to help facilitate the processing of the protein through the pore, but also they were able to read each of the individual amino acids and to read at longer read lengths. And so this is an important contribution to the development of single molecule nanopore-based approaches for protein analysis. This area of protein nanopores is really meant to bridge the tremendous work that's gone on in DNA sequencing using DNA nanopores. Unfortunately, the challenges of developing protein nanopores are substantial. First, protein nanopores have to deal with the incredible diversity of the amino acid letters in the alphabet. And so you no longer have all nucleic acids that are biophysically similarly in charge. You have amino acids that are small, that are large, that are positively charged, that are negatively charged, and somehow you need to process all of those through the pore. You potentially also have to deal with things like post-translational modifications. And in this paper, they specifically looked at phosphorylation. So beyond the biophysical diversity, you also have the challenge of how do I discriminate each amino acid from each other? The nanopore is mostly able to discriminate on size and a little bit on speed of processivity. So squiggles as they look like in the paper. And so in addition to the biochemical aspects of the work where they were able to build nanopores and process model proteins through them, they also had to build a machine learning framework in order to interpret and identify each amino acid. This really is an exciting advance for the field, and also shows just how far the field has to come before we're able to routinely do protein nanopore-based studies.
Yeah, I mean, I agree. This is a major jump forward in the field of nanopore sequencing. Maybe just take a step back. I mean, it's a crazy idea to think about it, that you would take a biopolymer, whether that's a DNA or a protein, and try to suck it through a pore in some form or shape, and then by just controlling or measuring the potential differences that's generated when you bring that biopolymer through the pore, that you can actually sequence it. Seems an insane idea. I mean, it's now standard that this is done, even commercial instruments for DNA. But as you described, the DNA is a different beast than proteins, particularly when you have to differentiate 21 different amino acids. So I think the previous work that has been done in this area, where there was various attempts to bring proteins through the pore by appending a DNA strand, for example, and suck that through a pore, or by using specific enzymes that would essentially guide those proteins through the pore. This is quite amazing that at this point the combination of machine learning and studying known sequences, and then combining that with a mechanism to reread the sequence multiple times, is really to the point where you're starting to see reproducible signal of these potential changes reproducibly for substantial portions of the proteins that are being read. So a major step forward, and I was actually quite surprised reading the paper earlier this year.
I think one of the things so when we look at the specific advances that enabled this work, one was clearly the use of the ClipX protein as the motor for driving the protein through. The other was the use of a very special set of amino acid sequence linkers that included a mix of a standard glycine linker, but also included a series of negatively charged amino acids. And so that helped push the protein, the model protein, through the pore. The other really key advance was the way that they went about reading. Historically, most nanopores would run the protein or the peptide through. They'd be able to read about on the order of 10 to 15 amino acids, and then the protein or peptide would have scooted all the way through, and you'd only get a single read. But in this case, they were able to first load the protein and then read it back. And then by using a sort of sloppy, loose handle from the linker, the protein would essentially slip and then run through the pore again. And so it would read through, it would slip, then they read through and it would slip, and they would be able to get the multi-pass aspect. And that multi-pass aspect was really critical in uncovering and deconvolving what each amino acid was. What they found is nearly doubling the accuracy in many cases of reading of each of the amino acids, because they were able to read it over and over and over and over again.
Yeah, correct. And I think another aspect which is very amazing in this part of the particular paper is that they actually went much further than just being able to read a sequence. They actually went ahead to show that even in closely spaced phosphorylation, sequence that have a closer spaced phosphorylation situations, they could actually read those out faithfully, which is basically a very big problem in the field, that people need to be able to resolve these, what we call proteoforms. And so I was quite surprised that was actually possible using this reread mechanism, because you would imagine at the speed of reading that happens in these pores that, the bulky phosphate group would just be not giving sufficient signal, but the traces actually show some very nice signals for these double phosphorylation events.
Yeah, I think also that the phosphoserine, phosphothreonine, at some level beginning to start to look like other amino acids as well. And so the deconvolution problem is extra challenging. And so it was impressive that they were able to deconvolve those. Other types of modifications, meristylations, methylations, unclear as yet how easy it'll be to read those. But I think one of the key other advances was just the read lengths that they were able to achieve, that they were routinely able to look at read lengths of 100 or so amino acids in some cases, or longer, even though these were, again, model proteins or fragments of proteins. So one of the proteins they looked at in the study was a fragment of A beta. They also looked at a fragment of titin, which titin, of course, is the largest protein in the world. And we're able to deconvolve or fingerprint which those proteins were. So while they weren't necessarily able to get letter level resolution from direct proteins, they were able to get patterns that they could then infer which protein they were looking at. And that's a pretty significant advance over previous work.
So does that mean we're going to see nanopores for protein sequencing or proteoform analysis very soon? What's your assessment?
I think there are still a number of key challenges that are left to be done. The first is, in this paper, they used a lot of very specific amino acid sequences as leaders, as intermediaries, as glue between individual amino acids that were being read. And so that sort of scaffolding and how one does the sample preparation to convert a complex mixture of proteins into something that can be naturally processed through the pore remains an open challenge. The other challenges, of course, are that real proteins are complexly modified with glycans or many other modifications. The extent to which those clog pores or disable pores is as yet unknown. And I think the other challenge, of course, is just how many reads are you able to get? How many protein molecules are you able to measure per unit time? The number of pores available, even on the largest nanopore systems, are still relatively small relative to the billions of molecules that are present within even a single cell. And so I think we are moving to a world where the Naval Lab and others have made just amazing progress on realizing this technology, but there's still some pretty fundamental challenges before this is routinely available.
Yeah, I would also say in terms of moving that to practice, not every, as far as I understand, not every pore that ends up in these membranes is actually functional. And so what you see is basically ideal scenarios. And so there's substantial work to be done to sort of make that a routine tool. But nevertheless, it's extremely impressive to see the resolution that I've been able to obtain and deconvolute on these signals. And it keeps reminding me, I mean, some of these signals are so hard to interpret as a human that basically machine learning is the only way to sort of get faithful readout of these, these types of signals. And so I cannot imagine any of these new technologies emerging without, appropriate machine learning algorithms developed in tandem.
One of the other things that was really exciting for me was just an open question that I had was how much of the protein traverses the pore at any given step. And is it 1 amino acid? Is it 12 amino acids? And that comes down in part to the time resolution of your sensor. And what they showed in this paper was that it was about 2 amino acids that traverse the pore per per each of their steps. And so that actually is quite good resolution. And again, in a multipass setting where you're able to go back and look at patterns over and over again, that's sufficient resolution. Ideally, you would like it to be that you had many temporal samples and your time resolution allowed you to go very slowly as you were reading, or go very slowly where you had uncertainty, and to have that kind of control. But the ability to have each individual pore sampling at different read lengths or different read times continues to be a challenge. Essentially, this is a massively parallel effort, and sometimes you don't have individual control of individual pores.
So given all the limitations that still have to be worked through, or challenges I should say, that have to be worked through, but one can imagine that these problems can be overcome, and that a basic system could be generated in the future to do these type of protein analysis. What kind of applications are you thinking of?
I think the scale of applications is really going to correlate directly to the scale of reading. So for instance, a clear aim of the authors is to look at proteoforms and to be able to read large segments of proteins where you're able to say, oh, this is a triply phosphorylated or quadruply phosphorylated protein. Even in looking at individual proteins derived from pull downs, for instance, where you don't necessarily need the total number of reads that you do for broad scale proteomics analysis, to be able to uncover or discover new proteoforms or new proteoform families would be a really tremendous application of this work. One other aspect they're looking at is fingerprinting. So can I fingerprint a set of proteins? Even if I'm not able to deconvolve the total proteome, I might be able to look at lower resolution quickly and say, oh, okay, well, this protein is present or not present in this sample. And that could have uses both for looking at targeted degrader studies to see if something is there or present, there or gone. That could have applications in looking at biodefense applications to see if potentially a protein toxin is present or not. So there are many applications where you don't necessarily need the full breadth of the proteome, but having letter level resolution for looking at variants or particular strains or PTM proteoform patterns could immediately be applicable, could immediately take advantage of these kinds of tools.
Yeah, I was thinking of similar fields of applications where you need to study individual proteins that can be relatively easily isolated through a precipitation or simple sample prep in more detail, because they're for example, are allergens. And one could imagine that this technology lends itself to miniaturization. So this type of high sensitivity single molecule analysis can actually be done outside of laboratories. And there's a number of applications, I think, where the ability to measure particular proteins or proteoforms would be absolutely useful. I mentioned the allergens, but there's also applications in authentic food or other areas.
One last point I'll make is that I want to make sure we don't lose the thread, is that this use of the ClipX mediated protein unfoldase as a key motor for a nanopore, I think is a very important contribution. What is the enzyme that drives a nanopore is the question when developing tools like this and approaches like this. And I think the authors showed very convincingly that this is an excellent approach, and that their general strategy for how to build and design a protein nanopore has a lot of promise.
So maybe as a Duncan experiment, when I saw this paper, I thought, I wonder whether you could create a giant funnel on top of it, where you put a single cell in there, and then you bust the single cell up, and then you let all the single proteins that are sort of falling into the funnel suck through hundreds or thousands of these pores, and literally sequence the entire single cell. Obviously, it's not something that could be done anytime soon, but I think that would be the perfect setup to sort of literally look at every single protein molecule in a single cell, a dreamlike experiment.
Well, since we're going out to the far future, there actually are these amazing nanotechnologies now that are essentially straws. So they're nano straws, often made of carbon nanotubes, that can perforate cells, and they're used as sippers to sample the interior of cells. And so if you were to combine a carbon nanotube sipper straw with a nanopore technology like this, could you perhaps realize your vision a little sooner?
Possibly.
Just to sum up, this work by the Navala Lab is really a foundational work for extending what's possible in using protein nanopores to study and sequence individual protein molecules. And their advances in both their biochemistry and their use of a ClipX unfoldase, as well as a CSG nanopore, really lay a foundation for many future exciting advances. They also really were able to identify each of the 20 amino acids, as well as potentially phosphorylated versions of some of them. And so exciting work by a lab definitely to pay attention to, and really excited to feature them on our episode.
Yeah, congrats to Keisuke and Daphne for the excellent work.
Thanks so much, everyone, for joining us on this special end of 2024 episode of Translating Proteomics. With these 3 papers that we've shared, we have hardly scratched the surface of all the excellent proteomics research published in 2024. I encourage you again to share your favorite publications from the year in the comments, or by sending an email to translatingproteomics@nautilus.bio. Here's to an exciting year in proteomics, and we're sure there will be even more excitement to come in 2025.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.